Welcome everybody. Please come on in. Find a seat up close to the front since the screens are very small. So my name is Jeff Shima. I am a professor of ecology at Victoria University of Wellington in New Zealand, and it's a great pleasure to come up this way to speak to you guys. I'll be talking about some work that we've been doing over the last 8 years or so. We're quite excited about it. Speaking on behalf of my colleagues Suzanne Alonso, Eric Noonberg, Craig Osenberg, and Steve Swear. And so I'll be talking to you a little bit about how we think that reef fishes might be spawning on a lunar cycle for very strategic benefits and potentially able to manipulate the sex of their offspring by virtue of when they spawn. Okay, so a bit of background about reef fishes for those of you who are unfamiliar with this group of animals. Nearly all reef fishes have what we call a bipartite life cycle, where the adult stage that we see on the reef are easy to see and study. They make larvae that develop offshore in the pelagic for a period of time. Very difficult to study those individuals, and importantly, that's where a lot of the action happens. There's a lot of mortality that happens out in the pelagic that drives the population dynamics of these organisms. I'll be speaking mostly about the 6-bar wrasse, the Thalassemia hardwicki, which is what you see here in the picture. It has many of the characteristics of an archetypal reef fish. It has this bipartite life history. One of the features of Thalassemia and the species in general is that it's highly iteroparous. So we can see evidence of spawning activity year-round, nearly every day of the year. Not necessarily by individuals, but as a population, there's spawning activity happening all the time. The product of those spawning activities are pelagic eggs that hatch into pelagic larvae that develop over 47 days and then return back to the reef. And most of the successful settlement, most of the successful returns back to the reef happen at night on the new moon, or very close to the new moon. And so there's a very strong lunar rhythm in settlement patterns that we see. So there's like a good settlement window in which settlers want to be arriving. We think that the reason that underlying this is that there's reduced risk of predation during these dark nights when there's no moon. So you can avoid being you can settle under cover of darkness and avoid being seen by visually hunting predators. So this sort of poses a bit of a conundrum. We've got a species that as adults they spawn kind of any old time. They have a period of time for their larvae to develop, but there's kind of one period in the month which they want their babies to come back. Okay. Another feature of the system is that it's got really cool sexual maturation sort of things happening. It's a diandric protogynous hermaphrodite, so first maturation can be either to the primary male or to a female stage, and that's driven by the local environment, those population density and things happening there. So that's an important feature of that system. And so potentially parents can manipulate when their offspring arrive back to the reef and shape the probability of them maturing to male or female. Okay. So kind of one of the first observations that we've made, I'll kind of walk you through this issue, why we think it's parent spawning at the wrong time. But first I want to orient you to these two panels here and what you see on the x-axis. So you'll see a number of panels that have 0 to 30 on the x-axis, and here I've just shown you little moons. This is to remind you that this is a lunar calendar. So a lunar cycle is 29.53 days. And I'm representing that from 0 to 30. So 0 and 30 are effectively the new moons. Halfway through the cycle is the full moon. So it goes new moon, waxing moon, full moon, waning moon, back to new moon. So the panel wraps from the left to the right side. Okay. And so we've quantified egg production over replicate lunar cycles. We've gone out every 2 to 3 days, and we've counted the number and sizes of individuals that are engaged in spawning activities over a 6-month period. So we have 6 replicate lunar months of spawning observations. And we've also sampled the products of those spawning activities over that same 6-month period, collecting the settlers that come back to the reef. And we can reconstruct the birth dates of those individuals by looking at their otoliths, their ear bones. And so we can count the number of rings and determine when that fish was likely hatched from its egg. And so this is the sort of pattern that we see for egg production on a lunar cycle and birth dates of settlers on a lunar cycle. And what you can see is their mismatch, right? So most of the reproductive effort by adults is happening at the new moon, and the least amount is happening at the full moon. But when we look at the birth dates of settlers that have survived that pelagic stage and come back to settle, you see the inverse pattern. Most of the settlers are actually born on the full moon, and relatively few are born around the new moon, when most of the adults are spawning. And if you estimate selection based on those two patterns, you see very strong evidence of a selection that should be favoring adults to spawn more than they do at the full moon and less than they do at the new moon. So this is why we start with this observation that what's going on with the parents, why are they seemingly spawning at what seems like the wrong time, if the goal is to maximize survivorship of larvae. Okay, so it seems like a silly strategy. So we've thought a lot about this, and I'll show you kind of a graphical representation of our hypothesis, which is effectively we're looking at a timeline of spawning and settlement, and overlaying that timeline is a lunar cycle. So we think the lunar cycle is really important in the system, and we have a highly iteraparous fish that can spawn anytime in the lunar cycle. Some of those spawning times will produce larvae that, if they develop for 47 days, will arrive back to the reef at a good settlement window, which I'm representing by this little window-shaped thing on the timeline, corresponding to a new moon. It's dark at night, fish can settle back to the reef and hopefully avoid being eaten. Fish spawn at the full moons, if they settle 47 days later, they're likely to naturally spawn, settle at a new moon. Fish spawn at a new moon, if they settle 47 days later, they're likely to naturally spawn at a full moon, which we think is a bad time. So one of the parts of the hypothesis that perhaps offspring can compensate for bad birth dates. Perhaps they can shorten or lengthen their development times to get around this problem of being born at a bad time. So that's question one, can they compensate? And then we think that compensating comes with consequences. So what are the consequences of extending your development time or shortening your development time? That has implications for the size at which you come back to the reef, the condition at which you come back, maybe what sort of areas you end up in. So we expect there might be consequences to this, and we think that those consequences, because we have a system where sex change or sex maturation is determined by the environmental conditions and the traits of those individuals, that those consequences can translate to sex change. That's kind of where we're going with this. And part of our original formulation of this was that we proposed this interplay between spawning decisions of parents and the ability of offspring to compensate might lead to a dynamic feedback that basically supports the evolution of extreme iteraparity in these species. Basically, adults can spawn whenever they want because the babies can sort it out. And perhaps this paves the way for a reproductive tactic in terms of spawning can be somewhat strategic if you want to make a male or a female. So first question, can offspring compensate for a bad birth date? This is the distribution of larval durations of development times. The 47-day mean is shown in white, and then increasing shades of orange are shorter and shorter larval durations. Increasingly dark shades of gray are increasingly long larval durations. So we can ask how the distribution of larval development times varies as a function of one's birth date. And so that's what this panel here is showing. So again, this is showing the panel on your right is showing the lunar calendar, and you see a series of stacked bars showing the distribution of larval durations for fish born on, say for the sake of example, on the new moon. So what we see for fish born on the new moon is that nobody goes 47 days. Fish are largely either increasing their larval duration or greatly decreasing their larval duration. Effectively, the distribution of birth dates for fish, or distribution of development times for fish born on the new moon, is bimodal. So a peak at sort of the low end and the high end, and nobody's going the average. As we move across, we see a lot of gray here, and so fish in the waxing moon are largely kind of going longer. Fish around the full moon are largely going kind of the mean, and fish around the waning moon going back to the new moon are largely shortening their development time. So effectively, they're kind of making adjustments to their larval development to either shorten or lengthen it to hit the closest available new moon settlement window. Okay, so we think this is suggestive of adaptive plasticity in development time. It doesn't explain why fishes are mostly spawning on the new moon. So let's take a look at how birth date shapes early development. Okay, so what I'm going to show you now is how fish larvae grow as a function of the lunar cycle. Again, I'm using otoliths to reconstruct the growth patterns of individual fish larvae that we've sampled, and I'm showing this as an estimate of residual growth. So I'm removing sort of age and size related effects on larval growth rate, And what I'm going to show you are a series of residuals. So things above that dotted line are fish that are growing faster than average for a given size and age. Negative residuals, fish are growing slower than average for a given size and age. And if we just look at how larvae are growing as a function of the quarter of the moon in which they were born, for the first 25 days of their larval life, there isn't a lot of variation. Everybody's kind of growing the same sort of way. After about 25 days, we start to see some interesting patterns emerge from these residual growths. We see these harmonics happening. The peaks of those harmonics and the troughs are separated by 7 days. And effectively, this is a lunar rhythm. So this is, and the reason why these are offset is that fish are starting their journey at different stages of the lunar cycle. If we align these same data instead to a lunar calendar, this is larval age, but if we plot it on a lunar calendar, this is what you see. So a pretty remarkable lunar rhythm in larval growth. Worst growth happens at the first quarter moon or the waxing moon. Best growth happens at the last quarter or the waning moon. That's a 24% difference in growth rate between the worst and the best time. So a really big effect on growth happening as a function of the lunar cycle. Coming back to sort of why that might matter for traits of fish that settle, we think new moon birthdays have some advantages. So one of the advantages is that if you were spawned on a new moon, by virtue of when you enter these sort of periods of good and poor growth, you actually get 2 bumps in growth, as opposed to all the other lunar quarters that only experience 1 beneficial period of growth. Moreover, you end up experiencing your second bump in growth just as you're coming back to the reef. So just as you're settling, you're growing better than everybody else. Okay. So some consequences for those larval experiences that vary with the lunar cycle are that fish that are born at the new moon, so the green panels here, are settling a bit before all the fish that are born at other quarters of the moon, so they gain what we call a priority effect. So they're getting to the reef first, getting to pick the best sort of places. By virtue of extending their development to hit that settlement window, predominantly they're settling at a larger size, and they're settling at an older age. And again, they're arriving to a reef at a time when the spaces are largely empty and they can pick the best sites. So we get these fish are arriving to these offshore sites in the system, which we know from other work are higher quality sites. And they're also slightly more likely to settle in social groups. And all of these traits we know, at least from another species of thalassemia, form the basis for environmentally determined sex in these species. So fish that settle into those conditions are more likely to mature as males. Okay, so we think we see increased probability of maturation to the male phenotype. Okay, so very quickly sort of working through. So evidence for strategic spawning. So this is the final sort of data slide. So what we're seeing here is this is effectively the data I've already shown you. Most of the spawning activity is happening at the new moon with the least at the full. We see that inverted for settlers. We see that further inverted for juveniles. So there's kind of a mismatch with juveniles. But if we look at the otoliths of adults that mature to male, we start to see a pattern of birth dates that closely resembles what we see for the spawning pattern in females. So, and again, sort of a high proportion of primary males were spawned at the new moon. A high proportion of females appear to be spawned at the full moon. So kind of very quickly revisiting this hypothesis, we think offspring can adjust for bad birth dates. We think this shapes the conditions for sex differentiation, and we think this helps to reinforce iteraparity and might reinforce opportunities for sex determination. Okay, so I think I'm out of time, so I will just go straight to the acknowledgements. So thanks.
Low. Ha ha ha. Okay, should I start? Okay. So, afternoon everyone. My name is Chedhawat Chokechaipaisarn, or you can call me Tim. Yeah. I'm a PhD student from University of St Andrews, UK. I work with Andy Gardner, and here's my talk: Density-dependent dispersal promote female bias, sex allocation and reduces sex ratio conflict in viscous population. So my works revolve around 2 words: sex allocation and viscous population. So if you imagine that you are able to produce many offspring, how many male or female you would want to. And that's when sex allocation theories comes in handy. So sex allocation apportionment of reproductive resource to male versus females. But in this case, when I say sex allocation or sex ratio, I'm referring to the proportion of sons or the number of sons per total offspring. So by initial response, you might say, well, the number of females and males should be equal, right? And yeah, because both male and female, they both make equal genetic contribution to the next generation. And that's the principle behind the theory of equal investment in sex allocation theory. But that's not always the case. In 1967, Hamilton proposed a theory by imagining an infinite population subdividing into patch. Each patch starts with n mother. And these mother produce offspring, they mate locally. And after that, all the female disperse. So by this life cycle, the optimal proportion of sons, or the optimal sex ratio, is depicted by this equation, n minus 1 over 2n. So here, if the number of mother is a large number, the sex ratio will be closer to 0.5 or equal sex ratio. But if the number of mother is a low number, like a small number, sex ratio will be less than 0.5 or female by S sex ratio. So what does this mean? It means that when we consider the relatedness between male offspring, it is better to avoid the mate competition that happens between male kin. And that's why female by S sex ratio is evolved in this case. And this is first ground-breaking rule in the sex allocation theory. So theorists might say, well, Hamilton has already considered the relatedness between male offspring. How about we consider the relatedness between female offspring on top of that? And that's when they began to imagine the situation called viscous population, a population structure where dispersal is low and create a local concentration among relatives. So they basically limit the female dispersal on top of the Hamilton's model. And does this do anything? The answer is no. In 1986, Baumer and Frank, they find out that when limit the female dispersal, yes, they increase the relatedness between females, which promote female by sex ratio, but it also intensifies the resource competition between female kin, which inhibits female by sex ratio. And these two effects completely cancel each other out, so there's no total effect on dispersal on the evolution of sex allocation. And this is a second groundbreaking rule in sex allocation theory. Dispersal has nothing to do with the evolution of sex allocation. And another key idea for my work is the similarity between sex allocation and altruism, especially when you change from equal sex ratio to female by sex ratio. So by the act of decreasing number of your sons and increasing number of your daughters, in a way, you will lose the direct benefit from your sons. But in the very same way, you will gain the indirect benefit by decreasing the competitors for your male relatives. So in the kin selection points of view, the act of producing female by sex ratio is equivalent to the act of altruism. And that's why when we see that there's no effect of dispersal on the evolution of sex allocation, the very same phenomenon also happened with the evolution of altruism as well. And that's when people began to try to find a way to entangle this cancellation. And in 2022, Conwell and Gardner find a way to do that. They propose that if individuals are able to adjust their dispersal rate based on density, density-dependent dispersal will promote the evolution of altruism in the viscous population. But we don't know anything about the density-dependent dispersal on the sex ratio. And that's become my first question, how density-dependent dispersal impacts upon the evolution of sex allocation in viscous population. So I'll start by imagining the situation like in Hamilton's by a life cycle of infinite population subdividing into patches. Each patch start with n mother. Mother produce offspring and they mate locally. After that, some female disperse. And after the dispersal, female will compete for n breeding spot to be the next generation mother. and I transcribe all this life cycle into the fitness function, which is the total fitness of making sons and daughters. And I'll use this fitness function to calculate the optimal sex ratio that maximizes the fitness through two routes, the individual routes or direct benefit, and the related neighbors route or the indirect benefit. And I'll derive this optimal solution or optimal sex ratio into different scenarios. So first one is a scenario where female cannot adjust their dispersal rate based on density. We call this density-independent dispersal scenario versus density-dependent dispersal scenarios. And here's the proposed mechanism that would happen. So imagine there's two patches with different density. In the viscous population, for the first scenario, some portion of females will be moved out from patch. And by this case, the relatedness will increase, but it also intensify the kin competition, which completely cancel out, so there's no effect of dispersal on the evolution of sex allocation in this case. But for density-dependent dispersal scenarios, so according to Presby and Taylor in 1990, the density-dependent dispersals will ultimately equalize the number of non-disperser to be equal throughout of the population. And that's why when two effects arise, now the kin competition effect will be relaxed and the cancellation breaks down, allow dispersal to have an effect of the evolution of sex allocation to become more female bias. So here's the result, and I derived this result in three genetic systems: haploid, diploid, and haplodiploid. So as we can see, the solid line represents the sex ratio for haploid and diploid, and dashed line is for haplodiploidy. And when I say that, I want you to look at the impact of dispersal or limit dispersal. I want you to look from the right to the left of the graph, where dispersal from full become more limit. So here, from right to left, the sex ratio, optimal sex ratio in haploid and diploid is basically constant throughout the dispersal range, so that there's no effect of dispersal here. While in haplodiploid, there's a really tiny effect of dispersal, which come from the inbreeding in this case. So there's no effect of dispersal on the evolution of sex allocation in this case. But for density-dependent dispersal scenarios, we can clearly see that from right to left, dispersal become more limit or viscous population, sex ratio will become lower or more female bias sex ratio in this case. So we can clearly say that density-dependent dispersal promote female bias sex ratio in the viscous population. However, this result is based upon the assumption that mother is the one who has full control over sex allocation. And how about when father is the one who controls the sex allocation? What happened there? So this becomes my second question: how density-dependent dispersal impacts upon the evolution of father sex ratio in viscous population. So I go back to the life cycle again. Now I throw the handle to father and see what happened here. So from a result in haploid and diploid, in the viscous population, I found out that father is agreed with mother, so they exhibit the same sex ratio optimum, so there's no conflict here. But in haplodiploidy, there's a sex ratio conflict happen in this case. So here's a result in haplodiploidy. So from now, the solid line will represent the mother sex ratio optimum, and dashed line will represent the father sex ratio optimum. And here we can see that father also always invest on production of daughters mainly, because they only contribute their genetic to their daughters. So they exhibit a greater female bias sex ratio than mothers. And this also happens in density-dependent dispersal scenario. But what changed? It's when from dispersal become more limit, or in the viscous population, we can see that in density-independent dispersal scenarios, there's no change in in the different in sex ratio optimum or degree of sex ratio conflict, while in density-dependent dispersal scenarios, the sex ratio conflict is greatly reduced in this case. So to summarize, in 1986, Baumer and Frank say that there's no effect of dispersal on the evolution of sex allocation. In my first work, I say, no, it's not. Dispersal can have effect on the evolution of sex allocation if individuals are able to do the density-dependent dispersal. And by this consequence, it will align the mother and father sex ratio optimum and reduces the sex ratio conflict in haplodiploidy. And now the next question is, can we test this prediction? I might say yes, there are some haplodiploid species who has male influence on sex ratio, like in parasitoid wasps and spider mites. I work with spider mites, is a really good system. Okay, and we can also extend this prediction to, because there's similarity between the X-linked, what is it called? Yeah, the X-linked control and the haplodiploidy, and we can also test it in the diploid taxa as well. So by this, I would like to thank all the people in these slides, and thank you for your kind attention. Yeah.
I guess I'll ask. So you were modeling female dispersal. Do you think that if males were the sex dispersing, that the strategy would be different?
Yeah, for sure. Yeah, because I think because this one is based on the male stay at the patch, right? And if we change it into like a male dispersing system, it might, what is it called? The graph may flip a bit. Yeah, and that's what we expected to see, right?
I actually had the same question, but I kind of want to follow up on it now. Do you think that that might give you an opportunity, because there is diversity where you have some female bias dispersal and some male bias dispersal, like might that be another opportunity to test some of these predictions and see if you get the kind of predicted change in sex allocation based on which sex is dispersed?
Yeah, that might be interesting to do, but what more? I don't have any answer on that on top of my head. I'm really sorry. Yeah.
We have time for one more quick question,
All right. Thank you.
All right, thanks everyone for coming. I'm Jay Gallagher. I'm currently a postdoc at UC Davis, but today I'm going to be talking about some ongoing work that I've been doing with my PhD lab in Robin Tingeltel's lab at the University of Denver. So one of the big questions that has always interested evolutionary biologists is how populations have changed over time. Now we can go into nature and see what a population looks like now, but not necessarily what processes got us to that point. So what happens during this black box, such as the mode and tempo of evolution, has been a hotly debated topic in our field since its inception, as we all know. Often how we see evolution taught in the classroom is that it occurs through incremental changes that build over many generations. Alternatively, evolution could proceed in more like a pulsed manner, with bursts of new variants arising rapidly, many of which don't make it and die out, followed by periods of relative stasis. Essentially, evolution could be throwing a bunch of spaghetti against the wall and seeing what actually sticks, a lot of it which doesn't. Unfortunately, studying this black box is challenging because we're often only looking at a snapshot of the evolutionary process. Comparative studies using, say, fossil data are great, and they can show us what has changed, but not necessarily how we arrived there, forcing us to make assumptions about a population's evolutionary history. This really stresses the importance of long-term, unmanipulated studies of populations that occur in nature. In particular, it would be really great if there was this system that had rapid generation times and was actively evolving in a way that we could track in real time. Well, fortunately for us, today I'm going to tell you about our system where this appears to be happening, and we're using it to better understand the evolutionary process. So male Pacific field crickets produce song by rubbing their wings together, and these wings are created by specialized structures that determine the acoustic properties of their song. I have some really important ones shown up here. Now, they sing these songs to attract females and entice them to mate, but in Hawaii, this also attracts this deadly parasitoid fly named Ormia that uses the song to locate its prey, killing the singing male and creating this really interesting conflict between natural and sexual selection that's unique to Hawaiian populations of these crickets. Over the past 25 years, we and others have observed several new morphs across Hawaii that each have either lost song, as in the famous silent crickets, or produce new songs that help protect them from the fly. Now note, none of these alternative morphs were ever found in sampling prior to the year 2000. Importantly, these new songs are due to modifications of morphological wing structures, and we know that these structures are integral to sound production. For example, if we look at one of these new morphs under the microscope, see my cursor? No. So if we look at the rattling wing, you can see that there's been this modification to a structure that we call the file. And we only notice this under the microscope. So in particular to our discovery of rattling, which, by the way, is morphologically indistinguishable from ancestral wings without using a microscope, we started to investigate these wings more closely. And what we found was a lot more variation than we had previously observed. Listen, I've looked at a lot of cricket wings, and I can tell you some of this variation is super, super weird. Like duplicated and looped files, veins in places they're not supposed to be, entire structures that are missing that are like on all cricket wings. And it seemed that this new variation was more prevalent to me than it had been in prior sampling trips just a few years earlier. So is it possible that variation has actually increased in such a short amount of time? If so, such a rapid burst of new variation might paint a picture that looks more like pulsed than incremental evolution. Well, luckily for us, our lab does repeated field work in a set of locations across Hawaii and other islands, so I've been able to take advantage of that sampling to piece together data to help us answer this question. Also note that all the sampling was done using methods that don't bias collection of certain male morphs over others, so we could use these data to figure out what variation was at a given place at a given time. So we looked at wings under the microscope from 4 different populations across Hawaii that we had at least 2 sampling time points for over a period of about 5 years, which is around 20 cricket generations. We also looked at the wings of a non-Hawaiian population that we could use as a control. So we then cataloged any of this variation that we found in sound-producing structures that was distinct from this ancestral wing that I have shown here. So this resulted in 13 morphological traits that we recorded for a total of 330 males each. And these were traits like the presence or absence of a branched file, you can see over here, or an additional vein that's inside the heart, which is one of those important structures. Using these data, we can ask a series of questions to see if our system is currently experiencing one of these evolutionary pulses. First, how do individuals group phenotypically? Will the variants we observe cluster with described morphs, or is this something new? Second, has the proportion of novel males increased over time in Hawaii? So these are males that have non-ancestral features. Third, and similarly, has the proportion of just previously undescribed variants, so this means variation that has not yet been documented in the literature, increased over this time frame? And lastly, are there any variants that existed in the past in our sampling but we no longer found it across Hawaii at any time points? So the more yeses we get to these questions, the more aligned our system would be with a pulsed mode of evolution. So to answer our first question, I took everybody from all our populations over the whole 5-year collection period and performed hierarchical clustering to group males by morphological similarity. So what do we see? Well, what our clustering shows is that there are actually a lot of novel individuals. But what's really surprising to me here, as you can see, yes, it shows, you can see in yellow, a large number of males have features or combinations of features that have not been reported before. It's a lot of them. In fact, we actually see 38 unique combinations of wing traits across Hawaii. And remember, these are all changes to structures that we know are important in determining song characteristics. For a deeper dive into one of these undescribed clusters that we call Broken Harp, make sure to go check out Vanessa's poster tonight. It is poster A1, so it's literally the first one. You cannot miss it. All right, so to answer our question, yes, there appears to be a lot of previously undescribed variation in Hawaii. Now we're going to delve into our next 2 questions, which is looking at the amount of novelty and how that's changed over time. So first, we'll look at the proportion of all novel males by population. So this means any individuals with wing traits that do not fall into this ancestral morph cluster. So they don't have features like that. And this includes any of the already described morphs that I talked about. All right, well, as you can see, populations generally became more novel over this 4 to 5 year period, which, as a reminder, is around 20 cricket generations. Now you can see this purple line over here. This is a Manoa population on the island of Oahu. This didn't change, but this is unsurprising because our recent work has shown that as early as 2017, this was already completely one new morph called purring. So you can't get less ancestral if you're already completely non-ancestral. Okay, now this is really rapid change, but it could be driven by, say, 1 or 2 morphs that have increased in proportion over time. So it doesn't necessarily indicate that the amount of new variation is increasing. So for that, we need to look specifically at change over time and the number of individuals with previously undescribed wing traits. So this is excluding all these known morphs that are here. So what do we see? Well, it appears that the amount of previously unreported variation has also increased over time across Hawaii. What this all means is that this previously unobserved variation that we are starting to notice actually is increasing across Hawaii. We're not just seeing it because we're looking for it now. It was actually not as present just 5 years earlier. So all this new stuff I was showing you doesn't, it seems like it's actually like new stuff that we're seeing, which I think is kind of wild, but that's just me. All right, okay. So at this point, you might be thinking, no, wait a second. We're now like actively looking for all this, right? So maybe I've convinced you that this has increased over time, but maybe this variation actually does exist at some levels in non-Hawaiian populations, right? We just didn't see it before because we weren't looking for all these microstructures. Well, we can address this by looking at the results from our control population, which is from a non-Hawaiian island that does not contain the parasitoid fly. So what do we see? So that's it. That's the graph. So we found no deviations from the ancestral wing in the non-Hawaiian population, which is confirming that, yes, this burst of variation really does seem to be an exclusively Hawaiian phenomenon. Now, for our last question, we wanted to know whether any of this novel variation that we're now documenting existed in earlier samples, but we didn't find it at any later time points. So this is kind of evidence of those evolutionary blips. Here I'm showing you that dendrogram from earlier with endpoints representing unique wing phenotypes. So this is morphological dendrogram. So might any of these be evolutionary experiments, little blips, experimental forms that didn't last over time? Yes. So several phenotypes, including a whole little cluster over there, existed somewhere in the past in Hawaii, but were not found in more recent surveys. And note that all these male types are just the ones that grabbed enough of a foothold in the population for us to be collected. So there's undoubtedly more variants that did not accumulate enough for us to catch them at some point. In fact, there's actually an entire population on the island of Molokai where Robin Tingatella first discovered purring back in 2017 that we intended to have several time points for. But we were unable to get that because they actually went locally extinct during the observation period. And we know this population differed phenotypically from all others on Hawaii. So it seems like we might have even witnessed an entire population-level blip during this very, very short window of time. Putting these findings together, it seems that on all counts, yes, Hawaii is proceeding in a way that more closely aligns with patterns of pulsed evolutionary change. Will we even be around long enough to see something like speciation occur? Well, we don't know how this story will end, but we do know that if this evolutionary snowball continues, there will likely be even more variation that pops up in the future that natural and sexual selection can then shape. For now, the spaghetti is on the wall. Stay tuned to see what evolution has in store for us next. Thank you. Lots of people oh goodness, lots of people to thank, including a lot of people here. Yes, and you all. Yeah.
Jay,
John.
do you think this is like release of cryptic genetic variation, or is it new genetic variation, or is it all key? Like, what do you think is it?
This is just, yes. We don't know, but these are really, really, really good questions. I think because this is so new, like purring and rattling, these other things we discovered like a few years ago, right? But this is like, I analyzed this like 2 weeks ago, right? So it's like, so we just like haven't had time to figure out what's causing this. So yeah, so some of it might be plastic, some of it could be G-body. Like there's so much stuff going on, yeah. So I can't really answer that. Yeah.
Following a question, thinking about your future work, are you considering making experimental crosses between the different forms to see the inheritance of?
Yes, that is currently happening right now in the lab. Great question. Yes, exactly, because then we could see how much of the variation is really heritable, right? And that makes a big difference, yeah.
Yeah, unfortunately, the next speaker couldn't be here in person. Had some travel issues, so they have a pre-recorded talk that I'll hit play on when I'm told to. Oh, should I? Okay.
Thanks for coming to my presentation. I'm Kuangyi, and I'm currently a postdoc at U of Toronto. Today, I'd like to talk about a recent study with Maria Savidio about the effect of sexual selection on genetic variance and the lag paradox. For sexual selection via mate choice to occur, there should be direct or indirect benefits from mate discrimination. The direct benefits can include resources and parental care provided by males. And for indirect benefits, there are two major mechanisms. The first is the Fisher process, where females prefers a male bearing display traits tend to produce offspring that carry alleles for both the display trait and preference. So the offspring may enjoy higher fertility. Also, this process will generate genetic association between preference and male trait within individuals. The second mechanism is the handicap principle. When display trait is the only signal of the male quality, females showing the preference will produce more viable offspring. But under both mechanisms, the indirect benefits from mate choice will rely on the presence of genetic variation in male display trait or quality. However, sexual selection is considered to deplete genetic variation in males. And subsequently, due to the reduced indirect benefit, strong preference and exaggerated display trait is not expected to persist. Obviously, this is contradictory to what we observe in natural populations. In fact, as the figure below shows, contrary to the expectation, sexual traits tend to have higher genetic variation than non-sexual traits. And therefore, it remains a long-standing puzzle how genetic variation in male trait is maintained despite persistent sexual selection. This puzzle is often called the lag paradox. And note that this paradox only applies to the situation when males offers no direct benefits for females. And the word lack here is used because under situations such as lacking, sexual selection will be strong since females can choose from a large number of aggregating males. To resolve the paradox, scholars have proposed several hypotheses. The key idea in many of them is that realized display traits often depend on genetic conditions, and the individual condition is often determined by a large number of loci, and therefore have a high mutation rate and thus can maintain high genetic variation. However, the paradox itself remains a conceptual statement and has not received rigorous examination. So the question of our study is that when does sexual selection deplete genetic variation? To answer the question, we built a quantitative genetic model. For simplicity, today I'll only talk about the Fisher process model, but we found that the results are similar for a handicap principal model. Basically, our model considers two sex-limited traits, the male display trait X and female preference Y, and both assumes to be normally distributed. And we also assume that both the male trait and the female preference are subject to viability selection due to fitness cost. And the sexual selection process is described by the preference function psi xy, which is the probability that a female with preference Y will mate with a male with display trait X. And for the preference, we consider two types of preferences, the absolute preference and relative preference. Both are commonly used in sexual selection models. For absolute preference, females with preference Y mostly prefer males with display trait value X equal to Y. And for relative preference, females with preference Y mostly prefer males with display trait X equal to Y plus the mean phenotype in the male population. To model the cost of preference, we assume that when a female's favorite male phenotype is rarer among males, the female will suffer stronger cost, perhaps due to longer time searching for mates. However, we found that the results are the same for the two types of preferences. The life cycle of the model starts from the juvenile population right after reproduction, followed by viability selection on male trait X and female preference Y. And after that, sexual selection occurs. Since our question is to ask the evolution of genetic variance, we adopt the infinitesimal model, which assumes that the trait is controlled by many small effect loci. Briefly, the model assumes that for genetic value of a trait in offspring, the expectation is the average of its father's and mother's genetic value. And there's also a noise term due to genetic processes like mutation and segregation. Based on that, we can write out the genetic variance of the trait after one generation. If we look at the recursion of the genetic variance, we can see that sexual selection alters male trait variance through two paths. For the first path, it is that male choice directly changes variance of the display trait in males. And the second path is more complicated. Briefly, due to mate choice, there will be association between preferences in mated females and display trait in mated males. Also, we know that there is positive genetic correlation between preference and display trait within individuals. Therefore, as a result, there will be association between trait X in mated males and trait X in mated females, which is shown by the dashed double arrow line in the diagram. We can also do some analysis to see how sexual selection alters the male trait variance through the two paths. For the first path, which is a change of display trait variance in the male population by mate choice, we can see that there are two terms. The first term, which is colored in blue, is a reduction of variance caused by sexual selection. This is similar to reduction of genetic variation by natural selection. But there's additional term, the second term, that exaggerates genetic variation, which is generated by variation in female preference. Well, if there's no variation in female preference, this term will reduce to 1, so that sexual selection will always reduce variation in male population. When the exaggeration in the second term outweighs the reduction in the first term, mate choice will increase variance of display trait in males. And our analysis found that this condition requires that variance in female preference, Vy, relative to the variance in the male display trait, Vx, exceeds the critical ratio. As the figure below shows, this critical ratio can be low, especially when selection on preference is weaker compared to that on the male display trait. For the second pass, which is the covariance of display trait X between mated pairs generated by mate choice, it is always positive. And if there's no satisfaction, that is when the preference strength mu here is 0, then this term reduces to 0. And therefore, even when sexual selection reduces genetic variance in display trait via the first pass, overall sexual selection may still increase genetic variance of the male trait if the covariance generated by pass 2 outweighs a reduction in pass 1. Here, the figure shows the result obtained from precise recursions of genetic variance. And the color shows the genetic variance of the male trait at equilibrium. The x-axis is the strength of viability selection on the male trait. And with the expectation, as viability selection becomes stronger, the genetic variance of male trait will be lower. And the y-axis here is the preference strength, which controls the strength of sexual selection. Basically, the figure illustrates that there are two conditions when stronger sexual selection can increase male trait variance. For the first condition, it happens when male trait variance is smaller than twice the covariance between male trait and female preference, as the right region of the figure shows. As we can see from the figure, when viability selection is strong so that male trait variance is low, stronger preferences will always increase variance. However, when the first condition is not satisfied, for sexual selection to exaggerate male trait variance, it requires that first, variance of female preference relative to male trait is large enough, and second, preference strength is strong enough. As the left region of the figure shows, when viability selection on the male trait is weak, so that the first condition is not satisfied, in panel A, as the preference strength becomes stronger, the equilibrium genetic variance of the male trait will first decrease and then increase when preference strength becomes strong enough. However, in the right figure, panel B, where variance of preference is too small, stronger sexual selection will always reduce genetic variance of the male trait. We can also do a similar analysis for how sexual selection affects genetic variance in female preference. Again, sexual selection will alter female preference through two paths, but now both are indirect. The first path is that since mate choice directly alters variance of display trait in males, and because there is genetic association within individuals, mate choice will indirectly change variance of preference in the male population. And the second part is similar to that for the male trait, which is due to non-random mating and genetic association within individuals. However, we find that stronger sexual selection will always increase genetic variance in female preference, even on the parameter space where sexual selection reduces variance in the male trait, as we can see from the right figure. So in summary, our model shows that sexual selection can alter genetic variance directly and indirectly via multiple pathways, as the right diagram shows. Specifically, we find that for male display trait, sexual selection can exaggerate its variance as long as variance in female preference relative to variance in the male trait is not too low, and also the preference is sufficiently strong. We think that these conditions may often hold in nature. For example, the study on green tree frogs in the figure below show that there can be great variation among females in their preference for call frequency of male frogs. For female preference, we find that stronger sexual selection will always increase its variance. Overall, these results may partly explain the empirical evidence that sexual traits tend to have higher genetic variance than non-sexual traits, Also, it suggests that the paradox may not exist in the first place. In fact, our results suggest that it's under situations such as lacking, where preference tend to be strong and cost of mate choice is weak, that sexual selection is very effective in exaggerating variance in male trait. At last, I would like to thank my colleagues at University of North Carolina at Chapel Hill during my PhD study, and the EB postdoc fellowship and colleagues at U of Toronto. Unfortunately, I'm not able to attend in person to answer questions, but I'm happy to take any questions or comments by email. And thanks all for watching my talk.